Hello, and welcome to our presentation on electron microscopy. Electron microscopy initially originated with Hans Busch in 1926 with his invention of the electromagnetic lens, which allowed for the focusing of charged particles. This then paved the way for other physicists such as Ernst Rüsker and Max Knoll, which in 1931 created the world's first electron microscope at the University of Berlin. Rüsker further studied the electron microscope until 1933, when electron microscopes were able to obtain considerably higher resolution when compared to light microscopes. Electron microscopy is a field which continues to develop to this day, with Rüsker receiving the Nobel Prize in Physics for the invention of the electron microscope, in conjunction with Heinrich Rohr and Gerd Binnig for scanning tunneling microscopy.
When something is too small to observe with the naked eye, we turn to microscopy. Using magnification, we can see smaller objects or closer to the surface of larger objects. Whilst the amount of magnification can differ, generally microscopy can allow us to see things of size between 1 to 1,000 micrometers. But when something is smaller than that, we need a better source of magnification, a new way to view things. Enter electron microscopy, which uses electrons as a way to view either the internal parts of a very thin sample or the surface of a bulk of sample. The advantage of this is we're given extremely high resolution and extremely detailed images as a result. The wavelength of an electron is far shorter than that of visible light, with the wavelength of visible light being around 400 to 700 nanometers. An electron with an energy of 100 electron volts has a wavelength of around 2.2 times 10 to the minus 10 meters, so 0.2 nanometers, which is far smaller than visible light. Primarily, there are two types of electron microscopy: scanning electron microscopy, SEM, and transmission electron microscopy, TEM. TEM is used with a thin layer of sample and produces 2D images, and SEM uses the surface of a sample and produces 3D images, although it's important to note in both cases the images are in black and white and need to be colored artificially at a later date. Looking at the resolution of each technique, SEM has a resolution of between 1 and 20 nanometers, and TEM has a resolution of around 0.2 nanometers, which interestingly is 500,000 times greater than that of the human eye. These pictures were taken with a SEM device and a TEM device respectively, showcasing the resolution obtained in each case.
Theory of the instrument. So, electron microscopes operate similar to standard light microscopes, where waves are used in order to produce magnified images of samples. However, as electrons are subatomic particles, adaptations of electrons are required and need to be viewed as waves as proposed by Ohm and Schrödinger. This is the main theoretical basis behind electron microscopy, as the wavelength is based on the mass of the object, hence why electrons are used to view microscopic structures, as their mass is incredibly small. Once electrons are fired at a sample, there are various ways they can interact. Electrons can be transmitted, as seen in TEM, re-emitted, or scattered, commonly seen in SEM. As previously mentioned, SEM and TEM are the two most common types of electron microscopes. However, both use a different theoretical basis to one another. SEM uses primarily the scattering of secondary electrons from the surface to create a 3D image, while TEM uses transmission of electrons to create a 2D image. In the top right, there is a diagram displaying all the types of data a sample can emit, and the corresponding electron microscopy technique. And at the bottom there, you can see a diagram of how some electrons can interact with atoms of a sample to subsequently create an image.
So electron microscopy generally splits into two types, TEM and SEM. Of these two, TEM is similar to the traditional light microscope in that sample has, in this case electrons, passed through it, and an image is created as a result of the interaction of electrons with the sample. SEM, on the other hand, works based on firing a beam of electrons at a surface, and then the electrons that are scattered are detected and an image is generated from the detected electrons. There are similarities between the two types. Both initially have an electron gun, typically a heated tungsten or field emission filament. Electrons are accelerated down a tube, and all of this occurs under vacuum. This electron emission is focused into a beam through condenser lenses. The first focuses the beam on the specimen, the second focuses the electrons into a thin tight beam. At this point, the two methods differ. In TEM, the sample comes next and the electron beam is fired through it before being picked up by the objective lens before hitting a fluorescent screen and camera to produce an image. In SEM, the electron beam passes through some coils, then to the sample, with two detectors to detect the electrons that are scattered from the surface of the sample. From this data, images are generated, 2D images in the case of TEM and 3D images in the case of SEM. It's also worth noting that an extension of TEM, known as STEM, exists. In TEM, the electrons are shot perpendicular to the sample, whereas for STEM, the electrons are focused on a single point, known as a focal point. Much like in TEM, the image obtained is 2D, meaning the specimen appears flat. STEM can also be considered as a combination of STEM and TEM. So let's look at the two pieces of apparatus. Firstly, TEM apparatus. High voltage leads to an electron gun, which is the source of electrons to scan the sample. Then the two condenser lenses, which focus the electron beam onto the sample. It's important that the specimen is extremely thin so that the electrons can pass through it. Finally, the electrons pass through the objective lens and onto the fluorescent screen and is captured as an image. It's important to note here that this entire process takes place under vacuum, and the apparatus is fairly bulky in reality, which I'll discuss when we discuss the advantages and disadvantages of these equipments. For now, let's look at the SEM apparatus. This, as we can see, starts out the same as with the TEM, but diverges after the condensing lenses, since the sample for this method is at the bottom of the detector rather than in the middle of the detector. So past the condenser, there's a set of scanning coils, focusing the electron beam onto the sample. After being scattered from the sample surface, the two detectors detect the electrons and create an image based on the electrons detected. These two pieces of equipment have similar aspects to them, but ultimately are different, both in their operation and in the results upon the formation of the image of the sample.
Electron microscopy analysis. Before analysis can proceed, there are sample requirements that need to be considered. Using a dead sample when analyzing living organisms. Dead samples are used because the organism will die due to the vacuum pressure needed for electron microscopy. In TEM microscopy, sample decomposition can occur. This is because TEM microscopy can render a sample useless or decompose a sample. Due to where the beam is focused, the temperature can reach around 150 degrees Celsius. So a sample which can withstand this energy needs to be used. SEM microscopes use a lower energy beam, but this problem can still occur. TEM requires samples to be at least 100 nanometers or below to allow the passage of electrons through the sample. SEM samples do not require to be cut ultra thin, as SEM provides a 3D surface structure. Additional information. When equipped with X-ray detectors, electron microscopes are called AEMS for analytical electron microscopes, as it combines electron microscopy and X-ray microanalysis. Images in electron microscopy need to show contrast so that the regions of the image are distinguishable from one another. For example, in TEM, when analyzing crystalline structures, electrons are subject to diffraction by values from the Bragg equation. This will result in low contrast. Samples can be tilted in TEM, allowing images where the crystallographic planes are in a more favorable Bragg condition for more detail and information to be provided. Specific information is then obtained by sensors, and a computer will convert the data given into an image. The data received determines the image, e.g. transmitted electrons give TEM images, while secondary electrons and backscatter electrons give SEM images, and in the bottom right there is the Bragg equation.
Having had an extensive look at the operation and imaging of the types of electron microscopy, it's important to consider the advantages and disadvantages of these techniques. To start with, the main advantage of electron microscopy is the main reason for electron microscopy. It can view things many times smaller than can be viewed through the light microscope. By using electrons rather than light, we can view things 500,000 times smaller than with the naked eye. For example, bacteria can be viewed through these methods. Secondly, the techniques have been used widely across the spectrum of scientific disciplines. They can be used for biological analysis of bacteria and uncovering new species. They can be used in forensic analysis on samples obtained from crime scenes. They're even used in electronics in the design, manufacture, and research capacities. Microscopic analysis is becoming more and more important as industry advances and things such as technology become smaller. Finally, provided that the person who is using this apparatus was competent and well trained, extremely high resolution images can be obtained from analyzing the samples, which allows for better analysis overall. So to the disadvantages. First and foremost, because electrons can easily be interfered with by air molecules, the entire analysis process must be performed under vacuum. This is an important disadvantage both on the front of expense, to maintain a vacuous environment, and in the fact that certain samples, particularly living samples, require either significant preparation for analysis or cannot be used at all. Another consideration to make is that the images can only be obtained in black and white, meaning that colour has to be added after the fact. Whilst electron microscopy is fantastic for the areas of analysis it's used in, it's also extremely expensive to purchase and takes up an extremely large amount of space in the laboratory, which needs to be considered when acquiring one. Moreover, the apparatus itself is very sensitive to magnetic fields and vibrations from other pieces of equipment, so strategic placement of the electron microscope is necessary. The picture to the left shows an electron microscope in place in the lab, illustrating how big the apparatus is in reality. Finally, whoever uses the electron microscope has to be well trained to produce the high resolution and high quality images that can be obtained from it. This obviously means that a lot of time and resources are used in the training of people to use the electron microscopes. In summary, the main disadvantages are cost and space related, but training to use the apparatus is extremely important to consider as well.
Thank you for watching our presentation on electron microscopy.